All right, good afternoon. Good afternoon. Good afternoon. Thank you. All right, let's go ahead and get started. So I hope everybody is having a good summer of school. Just a few more days left. I know it's been busy, but I hope it's been good. And I hope you're also giving your feedback to the evaluators as they ask for information. And also at the end, we definitely would like more feedback about what you could do to improve further and how it was good for you or not and things like that. So that'll be very helpful. So today we are very delighted to have Professor Chelsey Simmons here, from University of Florida. Chelsey actually has a long history with our summer school. She was a student here 7 years ago, in one of the first ones that we held here that Professor Tahir Saif and Jimmy Shah organized at the time. So she's been a student, and now it was great to have her come back and give a lecture. She got her bachelor's from Harvard, and then a master's and PhD at Stanford in mechanical engineering. She also got a PhD minor in education, so she's very interested in the educational aspects of science and engineering. And then she also did a little stint as a visiting physician at ETH Zurich, and then now started as an assistant professor at the University of Florida. Works in biomems and cell mechanics, and will talk to us about her work. Thank you for being here.
Yeah, thank you. Thank you for the introduction. I have also prepared a visual introduction that will parallel what Rashid just shared with you. But if you'll notice, I did not have them print my slides for you. I had them print basically all of the pretty pictures from my slides, and you can and should annotate them yourselves. It'll keep you much more engaged while I'm talking and keep you from checking your smartphones. So hopefully you find that helpful. As we discussed, actually this was not mentioned, but I grew up, let's see if my laser works. I grew up in Gainesville, Florida, that's me. So I've been a Gator since I was little. You Illinois folks will appreciate, but orange and blue have always been my school colors. And then I did go on to do my undergrad at Harvard. And I tell you my history because I often get asked as brand new faculty, what did I do? How did I end up here? So I did my undergrad at Harvard where I majored in engineering. I took a semester off and did a co-op at DePuy Spine, one of Johnson & Johnson's orthopedic companies, where I spent some time in industry, and that was incredibly valuable. And I stayed at Harvard for a year after I graduated and worked in an administrative position. So I was actually doing fundraising for the university in science and engineering projects, where I was able to write the white paper for the Wyss Institute for Bioengineering and do a lot of fundraising for the Stem Cell Institute. So I got some insights into what faculty life was going to look like when I became a part of the machine that is the university. And that also really helped me decide I wanted to be faculty and contribute to those efforts. Before I started grad school, that's the dorm where everyone's staying, right? No? Maybe? That's the dorm they had me staying in when I was here 7 years ago. And those are some MIT colleagues who were here for the workshop that summer, who I am still in touch with. And then I went over to Stanford. My advisor should look familiar. Professor Beth Pruitt was here on Friday and spoke to you all. And if you still have those slides, I think my picture is on one of them from the work that I did with Beth. So I went to Stanford, graduated from there recently, and between Stanford and now, worked on my company. I think there we go. Worked on my company to commercialize some of the things that I developed during my PhD. And then went off to the mountains. That's me and my postdoc advisor, Viola Vogel at ETH Zurich. And so that was my path. And again, now I am back at the University of Florida, and that's Santi, who's actually here. Most of you have probably met him this week, and I don't know if you can tell, but our lab is orange and blue. So I got to pick the colors, so it has now officially come full circle. At the University of Florida, our College of Engineering is huge. We have 10 departments, 10 and a half. We have a sort of a sub-nuclear engineering department under materials. We're strongly ranked among graduate engineering colleges nationally, and we have a satellite facility at Eglin Air Force Base in the Panhandle. UF, as a land-grant college, also has satellite offices in every single county of the state, which I think is pretty cool. So we literally have a statewide reach. We are physically co-located with the College of Medicine, it used to be called Shands, now it's called UF Health, academic health center, so that trains all of our clinicians and has tons of clinical trials. We have a VA hospital in Gainesville that's connected to the university, and then we have College of Veterinary Medicine, dentistry, and as I mentioned, our food and agricultural science as part of the land grant mission is quite literally all over the state, and a lot of our animal science collaborators come from there. And Gainesville's awesome. Yes, I am biased because I grew up there, but we have great sports teams and a great arts and music scene, and surprising amount of natural awesomeness with beaches and hiking, and it's really a great place to visit. So come visit whenever you would like. The department itself, it's mechanical and aerospace, which was exciting for me because I know nothing about aerospace engineering, so I now have colleagues who know a lot about it. We have the largest department on campus, so we're actually larger than some of the other schools. And we graduate about 37 PhDs a year. We do research all over the board. That's someone up in, says Houston, we have a Gator. I don't know if you can read that. And there's some squid modeling. We have biomechanics. We have great design build teams. Our undergrads build submarines and boats and satellites that actually get launched into space. And it's really a great place to work. Last but not least, I was hired as part of this cell engineering initiative. Slide's a little hard to read, but Professor John Dobson, who's a magnetic nanoparticle person, is heading up this Institute for Cell Engineering and Regenerative Medicine, and we've been promised a new building within the next couple of years. So I say this to give you some background, especially for the undergrads, but also grad students. UF really is a great place to be. I'm very happy there, and I think my students are happy there too. So if you have any interest in UF for future studies, please do, or collaborations for those faculty that are here, please do feel free to be in touch. Okay, now we can finally get into the title of the talk and what I promised to talk about: mechanical design considerations for stretchable cell culture systems. So I included a video of muscle because muscle is a really good example of why we care about stretchable cell culture systems. The typical way to do cell culture is to take a boring plastic petri dish, put some cells in, dump some chemicals on it, do some arrays a few days later, and then we're very surprised as a research community, we're very surprised when the data that we find doesn't actually match what's going on in the body in real life. As engineers who think about material properties, it's actually not that surprising because nothing in the body is polystyrene and just sitting there. Everything in the body is moving, your brain is soft, your bones are hard, your guts are peristalsing to move everything downwards, your muscles are stretching, your heart is pumping, brain and heart and muscle are all firing electrically. There's a lot going on. And so the thrust of my lab is to try to replicate as many of these functions outside the body as we can in a reasonably high throughput manner. Okay, if we wanted to replicate all of them, we would use humans. Okay, but that's the whole point, is that we can't, so we want to try to replicate as many of them as we can. Okay, so cells can sense their mechanical environment. Hands up if you've heard the word mechanotransduction before. Only half? No, nobody said it yet at this conference? Yeah, you're sleeping through it? Okay, so when we typically think about biology, or when we thought about biology a long time ago, we thought about the biochemistry of cells, and we thought about how they sense chemicals, and those chemicals affect transcription and protein production and all of these other biophysiological things. But what we've realized in the last, specifically very carefully in the last 10 years, is that cells are also very good at sensing mechanical properties and turning those into traditional biophysiological signals. And so this process from an engineering perspective of turning one source of energy into another, this transduction, we can talk about that for cells as mechanotransduction, turning mechanical signals into biophysiological, biochemical signals that the cell knows how to deal with. And there are a lot of different things that mechanotransduction affects in biology. And so if you don't know your neighbor, please introduce yourself to your neighbor. And then go ahead and take about 60 seconds to brainstorm things in biology that mechanotransduction might affect. Go.
Let's go with the sound.
All right, my 60-second pie slice has filled itself out, so I'm going to stop you here. One of the things that I heard, which was nice as I walked around the room, is it's clearly an interdisciplinary group because everyone was saying something totally different, which was awesome. So I'm going to tell you what mechanotransduction means to me, but that does not mean your answer is wrong. I heard specifically on that side of the room talking about the senses. So at a very high level, how the tissues and organs integrate. My answers are going to be a little bit more on the cellular level. And so one of the things that affects at the cellular level, did anyone put phenotype, how the cells look? No? Where the subcellular proteins go? No? All right, so this is one of the big things about mechanotransduction. Again, at the very, very small level, almost at the molecular level, what we think is going on with mechanotransduction. But then we have tissue level stuff. Any groups think about the tissue level organization of cells responding? Maybe. Yeah, got a couple of head nods. All right. The protein production. So I kind of gave that one away earlier on, but the soluble factors that the cells are producing, and then, of course, just sort of overall transcription processes. Differentiation. I know I heard at least one differentiation. Hands up if differentiation, we came up with that. Yeah. So tissue differentiation, the different types of tissues we become in the body, or cells become in the body. And then certainly function. And so I guess that's a catch-all. I heard touch and smell and hearing. And yes, these are all responses to mechanical sound waves or mechanical force touching your skin. And the cells function differently given these mechanical inputs. All right, questions? You guys you should feel free to interrupt me, please do, as we go. Okay, yeah?
So some of these expressions of what's happening at a very small low level, function. What is that expression of?
So what is that an expression of? So I think so the way that I think about mechanotransduction is, you know those signaling pathways that certain biomedical folks will show at the beginning of their talk, and they'll be like, this protein is important because it's involved in 50 million signaling pathways, right? And so I like to think about the mechanotransduction and the mechanical effects as being a part of that pathway system and some sort of input that's affecting how all of those small molecules talk to each other. Is that sort of what you were asking?
For example, you mentioned protein.
Mm-hmm.
That would be part of the pathway.
Exactly, that would be part of a pathway to function, but also function, and this is going to give away my next slide, but function could just be opening and closing of an ion channel, right? So there are mechanically gated ion channels that if we pull on them hard enough, they open and they let stuff flow in and out. So that's more direct function, but a sort of secondary or tertiary function might be your heart cells contracting harder. Okay, when you pull on muscle, then it actually can contract with more force than if you don't pre-stress it. So, so given this mechanotransduction and how cells sense their mechanical environment and turn it into biochemical signals that allow the body to work, what proteins in the cell are likely mechanotransducers? All right, if you don't have a biology-oriented partner, you might want to turn further around to try to find one. But go ahead, we're gonna go for another 60 seconds and see how we do.
Angiotensin.
So it doesn't have to be a specific protein name. You can just think about like a part of the cell.
Cytoskeleton kind of thing.
Exactly. So cytoskeletal proteins are one really good example. Yep. There, yeah.
Let's say this is a child, but you know.
Yeah. Yep.
Oh, adrenaline. Adrenaline or adrenaline.
Okay.
All right, I'm gonna stop you here, though I heard some really good lists being generated. My rock stars up front are literally like listing names of proteins. That was way more specific than I was expecting you to come up with. The level I was thinking were like cell matrix contacts, right? There's a bunch of proteins involved in connecting the cell to, yeah, we had at least one group that came up with that. I think we had another group over there. Cell matrix contacts, there's a lot of proteins involved in those complexes that we know some of them are definitely mechanosensors, but it's a whole piece of the cell that's yet to be explored. Cytoskeletal proteins, I had a group over there. Anyone else? Cytoskeletal, yeah, back row. Cytoskeletal proteins, again, there's a lot of proteins in the cytoskeleton, and we're not exactly sure which ones are mechanosensors. We're not exactly sure how the transduction mechanism works, but we're pretty sure that they care about whether they're being pulled on or not. Cell-cell contacts. Do I have any cell-cell folks? Yeah, my cancer people probably came up with that one. The cell-cell, we think that that's involved in metastasis, and the cells deciding to split off and try to squeeze into the bloodstream is some sort of mechanical effect. The nucleus itself, do I have any people who really like lamins? No, one of my colleagues at UF is obsessed with lamins as mechanotransducers. So they're the proteins that actually surround the nucleus and are embedded in the nuclear membrane and are thought to also be mechanotransducers, and perhaps a much more direct link between the mechanical factors from outside and what's actually going on in the guts of the nucleus. And then there's all kinds of crazy stuff in the cell membrane linked to the cell membrane. So the intracellular complexes of the cell-cell contacts and the cell matrix contacts, but also integral proteins that are incorporated into the membrane. I mentioned mechanically gated channels to you, and then even quite likely the function of the peripheral proteins just floating around in the lipid bilayer are quite likely to be affected by the bilayer being stretched apart a little bit or relaxed a little bit. I'll leave this up for a minute so you guys can take notes. Questions as we're processing? Did I leave anything out that you came up with? No? Probably? Well, we had I heard from the front row, soluble factors that one cell type would express as a result of mechanical damage, then signaling to other cells that weren't sensing the mechanical stretch directly. So then there's also all of these secondary signaling factors that are probably related to mechanotransduction as well. Okay, so given this background for mechanotransduction and all of these things that are happening in the cells to the cells, let's take this back to what we're doing in the lab. So what we're doing in the lab is we're trying to recreate some of these mechanical conditions that you would find in the body, outside the body, to do the biology research. So the first mechanical feature that I focused on with Beth, actually with Beth Pruitt at Stanford, was mechanical strain. We wanted to replicate the stretching of cells that happens in the heart, in the muscles, in your skin, right? When you bend your elbow, it happens everywhere. So how do we replicate this strain outside the body for cell culture research? And to give you one, I think, exciting and compelling reason we want to do this, there's a group out of Harvard who's seen the lung on a chip before. Yeah, a few people? Okay, so this system, they took, I think you saw in the video, but they plated alveolar cells on one side that were intended to be exposed to air, and epithelial cells on the other side that were intended to be exposed to blood/cell media. And when they turn the vacuum on, they can make these cells breathe, right? They can make them mimic the breathing that's happening in the lungs. So what they did is they said, okay, we have this system that we can mimic breathing with. What happens if we try to poison it? And so they took silicon nanoparticles, which are known to irritate humans, and they just ran them by the cells with nothing happening, and the cells stayed there. This is reactive oxygen species. It's a sign of inflammation, how upset the cells are. When they just ran the nanoparticles by our triangles, there is really not an appreciable response from the cells. When they just stretched the system, the diamonds, again, not an appreciable response from the cells. If they had the cells breathe and they flowed the nanoparticles by, huge inflammatory response. I'll let that sink in. So this tells us two really important things. One, if we had been testing some new toxin, some new particulate that we didn't know how the body responded to it, we would say, and we had the cells just sitting there, we would say, oh yeah, big company, it's fine. Burn the plastic, dump it in the creek. We tested it on cells, everyone will be fine. Okay, but we would have been wrong because we tested it on static cells that were just sitting there instead of cells or mice or humans that were actually breathing. Okay, so it's safe for you if you hold your breath. On the other hand, what if we'd been testing an asthma drug? Okay, same thing, except the opposite. We would have said, oh no, this asthma drug doesn't work. But the asthma drug would have worked if we started breathing, which we're gonna do because we need oxygen. So again, really important motivation for creating these stretchable dynamic systems. Any questions about this paper before I go on? The references, everything I reference is on the last page of your handout, the full references, just so you know. Okay, so how do we recreate this strain in a way? I don't know if you saw this device, but this device is really teeny. It's complicated to make. It's complicated to operate. Plus, they published after I started my PhD, and I was already working on this problem. So how did I replicate mechanical strain in the lab? There are a few different ways to think about stretching cells. And again, I'm trying to focus on the design component here for you all, so I'll run through them. The first is this nice anisotropic, so an, not, iso, the same, not the same, in all directions, or uniaxial. People tend to use these words interchangeably. You grab onto something, you pull, it stretches. Okay, and so that's how the Ingber lab designed their lung on a chip. But there's also a lot of ways to do that in the lab by just grabbing onto something and stretching it. A little more complicated to design is your isotropic or biaxial, so the same in all directions or in two directions in plane. And so this is the design that we went with. If you pull a vacuum down, but you have a post supporting the membrane in the middle, then you get it to stretch in all directions. Or you can have an isotropic biaxial out of plane where it does stretch in all directions, but it moves a lot. And so there's a group that came up with a really cool system to create this kind of stretch using a Braille printing machine. So it has all these little pins that move, and so they used all of those little pins to stretch the cells. When I started my work, so after I attended this workshop and went back to Beth's lab and started looking at stretching systems that I wanted to build, I looked at what else was out there, and I saw that by and large, the stretching systems around there could only stretch at one strain at a time. Okay, you had to sort of set the machine, and it was going to stretch the cells X microns or millimeters for Y% strain, and that's what you were stuck with. They covered all different ranges. Here's one of Tahir's papers from a while ago. So at like a micron scale, all the way up to a nice 6-well plate from the Flexcell Corporation. But again, you could only get one strain level at a time. So as we're moving towards systems that are helpful and useful to biomedical researchers, being able to increase the number of parameters we can test at once becomes really useful. So that was really the goal of what I was doing, was to try to create a system that was sort of an intermediate size range, but you could test multiple strain levels at once. Does anyone not know what I mean by strain level? No? Everyone took our biomechanics for biologist class? Yes? Or is a biomechanics person? All right. I'm going to test you on it later, so now's your chance. Strain? Anybody? Okay. And then again, the design choices that I made was that we wanted a user-controlled waveform. We wanted to be able to input how the cells stretched. We wanted separate cell populations. Again, multiple strain magnitudes, and we really wanted it to be compatible with existing lab equipment. We didn't want to make everyone buy tons of new stuff. We wanted it to fit as best it could with existing lab equipment. So this is what we ended up with, a GUI written in C# that you could program your waveform into. It sent the waveform that you wanted to a microcontroller, which stored that information. And then the microcontroller controlled a vacuum and sensors in a nice closed-loop control. So if you said that you wanted a nice, slow sinusoidal pulsing of the membrane to mimic peristalsis in the gut, you could program that, and the system would execute that repeatably over time. We also used a 96-well plate. So looking at it from the top, it looks just like a 96-well plate, which means we can do traditional biochemical assays on the system by rescuing the cells, by taking the media. At each level of strain, the samples were independent. And then the big trick was having this supporting system underneath that allowed us to create multiple strain magnitudes in one experiment. Questions about the design, the overall design of the system? Not yet. Okay. Sorry. Environment in terms of CO2 and all that? This portion is intended to go in the incubator. So I don't know if you've ever checked. Most of the people I talk to are surprised by this fact, but almost every incubator I've ever seen has a hole in the top corner that's plugged by a stopper. Nobody ever knows it's there, but it is. And so you can feed these tubes through that hole and then keep the stretcher in the incubator and keep all of the electronics outside of the heat and humidity. Yeah, thank you for the question. Yeah?
How long do you keep them?
So our original testing of the system was sort of during the course of the working day. So it was 8 hours overnight, something like that. But we're working, Nick actually is working on experiments now where we're trying to build up to weeks at a time to look at stem cell differentiation. So it's much less a function. The electronics are great, for months. The bigger problem is actually keeping the cells attached to the silicone for that long. So we're working on our functionalization protocols more than the electronics. Yeah. Other questions? Okay. So the way that we created the different strain magnitudes in one experiment was by playing this geometry trick with the size of the post. So if you apply the same vacuum, but you have a wide post versus a narrow post, then there's different amounts of membrane for that vacuum to be acting on. So with a large diameter, there's not a lot of membrane. You get a small strain on top of the post. And with a small post, there's a lot of membrane, so you get a much larger strain. And hopefully you saw the video running. Let's see if we can get it to go again. Oh man, sorry. Okay, and so this is a video of the system operating. Those are dots that we micro-patterned onto the silicone membrane just for visualization. They're not cells, but you can see the membrane flexing as the vacuum pulls in a sinusoidal fashion. I did go on, as I mentioned before, I did go on to commercialize this system. So this is what it looks like once I asked professional engineers to work with it rather than me as a grad student. And this part right here is what goes in the incubator. And then this control box sits on top of the incubator. And then there's software to program the control box from your own computer. We also, and this has been Nick's baby this summer, is we realized that not every experiment we were going to do needed to be high throughput, given the wide range of mechanotransduction, mechanobiology problems out there, that some of the experiments we wanted to do we're going to actually want to watch while these phenomena were happening. So Nick has been working on a low-throughput stretcher, same idea, same design point that we want it to be compatible with existing lab equipment. So this is a tissue culture plate stage holder, right? So it's the insert that would fit in your microscope stage to hold a tissue culture plate. But instead of a tissue culture plate, we have this stretcher that's the exact same size. Was that a hand? No? Just stretching. Okay. And so again, we have, it's the same size as a tissue culture plate, so it fits into normal equipment. We have it computer controlled, so there's stepper motors that we can program. I voted MATLAB, but the students voted LabVIEW, so LabVIEW is going to win because they're doing it. And then we have our stretchy sample, which we can hold in place directly over the microscope objectives. Okay. Questions about the system? Yeah.
Does it affect the permeability of the membrane of the cell?
So the question was, does stretching the cell affect the permeability of the membrane? We think so. We're pretty sure it does. I suspect if you look in the literature, there are a variety of permeability questions that have been asked in a lot of different ways.
It affects the activity of the cell later on.
Yeah, absolutely. Absolutely. And then some of, we know, people have shown that things like adhesion molecules on the surface of the cell are upregulated or downregulated depending on the stretching regime. So that's not permeability of the membrane directly, but if you're talking about something like uptake of nanoparticles, right? The presentation of adhesion molecules or sticky sites on the cell could potentially be affecting that as well. So there are a lot of integrated questions to ask of these cells, which is part of the reason, again, coming back to design, part of the reason that we're designing these systems to be widely used to really integrate with normal systems out there, because as a lab, we can't answer all of the questions ourselves. We would love to. It would make me very famous, but we can't. We need help from collaborators. And so the more robust and simpler we can make our systems, the more collaborators we can have help us ask and answer questions like cell membrane permeability. Other questions? What I thought you were going to start asking was, are there differences in cell culture when you're using a permeable silicone sheet versus a not really permeable polystyrene dish? And there absolutely are. So using a system like this introduces a lot of new variables about environmental control and how to keep your cells happy. But again, it's a trade-off. It's an engineering trade-off. The question was, can I change the stiffness of the underlying material? Not for about 5 more slides. Yeah. so give me a couple more minutes. All right, so a real experimental problem that I've had along the way, and that Nick's going to have pretty much the second we get back to the lab, is that you can see the membrane moves in and out of focus. Okay, and that's due to this effect from Poisson's ratio. Did we talk about that in our biomechanics class at the beginning of last week? No, we've got at least one no over here. All right, so Poisson's ratio is the negative ratio of the stretch in one direction divided by the stretch in the other direction. And on your sheet, Poisson's ratio has units. It shouldn't. Please cross them out. It's a non-dimensional number divided by a non-dimensional number. It should be non-dimensional. So please correct that. But the question for you to work on with your neighbors is, if I have, we'll look at a simple problem, but if I have a membrane, okay, and it's, what did I make it? 50 millimeters long, 10 millimeters wide, and then 100 microns thick, and I stretch it 10%, how much does it move out of focus? All right, and I've given you enough information on the board to guess, but also everyone took the biomechanics class. So you'll have to sort of rack your brain, but we have 100 microns thick here. All right, so you get more than 60 seconds to talk about this, but talk about this problem with your neighbor for a little bit. I'll walk around. If you have questions, especially if you don't even know where to start, please throw up your hand. Yeah. Yes.
50?
Sorry?
Oh, so
Along the 50.
Okay, so that's long and that's wide. Okay.
Yeah. And the 50, and it's 10%, so it doesn't matter all that much because you can just, yeah. Yep. Yeah, so the length change is 5, but remember we're looking at the length change of the Z. We're worried about how much it's moving out of plane, yeah. But you're thinking about it right.
So we want the one in Z.
All right, so just to clarify, because I'm getting a little bit of confusion, this will help us move quicker. The long direction, the way it's being stretched is our axial direction. So this is our strain axial, which is the 10% that I mentioned in the problem definition. Yeah, there we go. So now we can plug and chug. Is that a question or a thinking? It's your thinking cap? Sorry?
Changing by 5%.
And changing by 5%, so how many microns is that?
If it's 100 micron thick, then it should be 9.
Yeah. Yep.
Is it 5 micrometers?
Yeah. He did, yeah. Do you have a question? Yeah, all right. I've gotten at least a handful of right answers, so I'll work through it for you real quick. But if we look at the Poisson's ratio, it's 0.5, which physically means that it's incompressible. Okay, so the silicone rubber, it's very hard to compress. Don't ever plug 0.5 into a computational model because it'll explode. But if you're doing an analytical model, 0.5 is okay. File that away. It'll come up like 2 years from now, and you'll be like, oh, that's what she was talking about. Okay, so we got 0.5 for Poisson's ratio, the negative sign, the strain in Z, which is what we're worried about. Okay, how much it shrinks out of view, and then the axial strain was 10%, so 0.1. All right, so we've got that. Our strain out of view is 0.05. Agreed? Questions about that? Does that make sense? It's probably more setting up the problem than anything else. Okay, so then strain, which I did not define for you, but hopefully you guys talked about last week. So strain is a change in length over original length, which should be 5%. So 5% of 100 microns is 5 microns. So change in length, 0.05 times the 100 microns. So we've got it moving 5 microns out of plane into Z. Does that make sense? Yeah? Other questions? We've got some excited right answers up front, which is great. All right, so now the follow-up experimental question is, how do we fix this? All right, it's moving out of view. I just told you that we designed a system to watch, right? We designed a system to stretch in one direction to watch real time. So in the next minute, brainstorm with your partner at least 2 ways to keep that membrane surface in focus at all times.
Yeah.
And then maybe you can.
Camera can follow.
Yes.
Camera is once omnifocus, right? It's called. But actually, don't worry about what.
Yeah, so there are some fancy, like I know Nikon makes one in their cameras. So yeah, there are some. So buying fancy equipment is usually a good answer to experimental problems. Yeah. So I'm sure that will come up.
Moving the stage.
Right, or moving the stage if you have a Z-motorized stage. Yeah.
The camera is lovely because you can vibrate the camera and move it.
Yeah. So it all depends on resolution in the end, anyways.
Mechanical property. So is it attached at the bottom here? So is it attached with?
It's actually I drew a bad picture, but these little bolts actually pin it down.
Okay.
Yeah. All right. Anyone want to contribute? They're good ideas. Everyone should, because there must be a million. You guys were talking like crazy. Yeah.
You could increase the depth of field.
Increase the depth of field. That is a great answer. So the depth of field with the 10x objective is greater than 5 microns. So if you're looking at it at low resolution, then actually it'll pretty much stay in focus. But if you crank it up to like 40x, the depth of field is more like 1 micron. So then we have problems. Buying a fancier objective Actually, in this case, we want a less fancy objective, so that we have a crappy field of view so we can see the whole thing, may or may not be a good solution depending on your application. Anyone? Yes?
We can move our stage.
We can move the stage, yeah. So some microscopes have an automated Z stage, so you can hack into the controls, and if you make the stage movement match the stretching, then you can track them in tandem. And so the NIH, what do they call it, micromanager software is quite good for writing code to hack in and take control of your microscope. Other thoughts, comments? Mm-hmm?
So an autofocus scope or a confocal scope, you can catch more of the Z planes.
Buying fancier equipment is usually a good option to most experimental problems. Yes, so a microscope equipped with autofocus is a great one. Another one is, remember the 5 microns was based on the original length or thickness of the membrane. Did anyone come up with making the membrane super, super thin? No? Because if it was, let's say, 10 microns thin, then it wouldn't be hardly shrinking at all in the Z, and it would stay in perfect focus. So I think we covered most of the territory. Anything else that I'm not thinking of? No? Okay. Good. That was some good brainstorming. I appreciate it. Thank you for humoring me. An advanced homework problem for those of you who want to think about the biomechanics more or are potentially designing your own stretching system is to think about the slightly more advanced problem of what happens if you're stretching in 2 directions. Okay, and so then the equation for Poisson's ratio is not so simple anymore. You need generalized Hooke's law. So the notes are on your sheet. You can do that as homework tonight. I know you don't have anything better to do, so you can play with that. All right, so back to our system. We had a great planted question earlier that was, it's great that you can stretch the surface, but can you change the stiffness? Can you change the materials? Because silicone has a very small range that you can actually tune the material properties within, and it tends to be stiffer than most biological tissues. So this was a question that Beth asked me a few years ago, and I said, sure, I'll figure it out. A few years later, and Beth and Rashid asked because this paper has special significance to me because I saw it for the first time at this meeting 7 years ago, and it changed my perspective on really what I'd been doing with my whole career, which at that time wasn't very long, but it still was very exciting. So what Dennis Disher presented to us 7 years ago that had pretty much just been published was that when he made hydrogels, and he made those Adam Engler made the hydrogels to match brain tissue, muscle tissue, and more or less bone, and they put stem cells down, keeping everything else the same. They got brain cells, muscle cells, and bone cells. So they changed nothing else except the stiffness of the substrate. And that totally controlled the fate of these stem cells. And so again, 7 years ago when I'm sitting in the audience, I think, wow, I've been trained as a mechanical engineer. I think about materials design. I think about stresses and strains for metals and plastics and bridges and bicycles. Now all of that is actually to apply to biology. Not in an abstruse philosophical way, but in a very direct mechanics affect everything about biology kind of way. So for me, this was very powerful, which is why I continue to show it. So again, moving forward, the question becomes, all right, we know strain is important, we know stiffness is important. How do we study these things independently and or together? Beth actually presented this, so I'm going to skip it, or at least skip to the end, skip to the punchline. Also, because I'm running out of time. When we culture cardiomyocytes on a soft surface, if we change that surface, so here's the elastic modulus, if we make that surface stiffer, our cells kind of sort of get stronger. Okay, so that's like saying, so here we hand the cells a 5-pound weight and they're bicep curling it all night long. All right, and they're happy and they're fine. We hand them a 60-pound weight, they do more work, right? They can still lift it and they're doing more work because it's heavier. But at some point, we hand them a weight that's just so heavy, they can only lift it up halfway. All right, and so they plateau. They max out at how much work they can actually do when they're contracting against these soft substrates. But what was really remarkable, and we did this in collaboration with Sean Palachek's lab, who is an Illinois graduate, and we found that when we put stem cell-derived cardiomyocytes down, they never maxed out, at least at the stiffnesses that we could test. They were supercells. We handed them heavier and heavier weights, and they kept pumping them. They kept doing the bicep curls. And so for us, this was important not only because it indicates that different cells respond to mechanics differently and that the mechanics really can affect function. And so to the earlier question, this is an example of mechanics affecting function. But it was an interesting insight into the new stem cell craze. And stem cells might be from patients who have certain diseases, but they're still not primary cells. There's still something different about them. So it's a bit of a cautionary note, but it's also exciting because we now have a model of something different that we can compare to the primary cells to try to figure out what the heck is going on. Yeah?
So what do you think is causing that increase in range?
That traction force microscopy is really hard to do. So I think in biology, there's tons of variation, especially in single cell experiments. But traction force microscopy in and of itself involves multiple levels of computation. And so the experimental acquisition of the pictures and then the calculations and the statistical optimization, I think you're introducing a lot of uncertainty and lots of spread into the calculations as you move along. Is that a satisfying answer, or should I keep talking? Oh, I see what you're saying. You're saying just like, why does it keep? So the short, I don't have a good like statistical high-level explanation. I think one of the practical explanations is the cell spread area is a lot more consistent and small at the lower stiffnesses. So I think you just have more variation in how the cells look at the higher stiffnesses. So if you have more variation in how they look and how they spread, then you're probably going to have more variation in how you do the calculations. We'll buy that, right? Okay. Yeah?
So why those particular stiffnesses?
Oh, so I did skip over that part, but we'll go back to it because I think it's useful. Okay, because these stiffnesses represent real developmental and disease states. So they represent baby heart tissue, adult heart tissue, and diseased tissue. Thank you for asking. And so then this is a picture of how it works. And again, you can see how hard it is to see the cell. It doesn't actually get that much better when you're looking at it at your computer screen. It's still hard to see. So that's one potential source of error, is just having to trace the outline of the cell by hand. And so then you're like, oh, put a label on it. Okay, but as soon as you put a label on it, you're now affecting new things and making it harder to do, or easier to do, it depends. But every step you add into the system introduces new uncertainties. Yeah? This is a single cell, and it's that shape. So again, it's a little hard to see, but yeah. All right. So hopefully I have convinced you that combining stiffness and strain is super important. So how did we do that? How do we do that, continue to do that in the lab? Well, we take silicone, so PDMS or actually commercially available silicone, an FDA-approved silicone, take your pick, and we treat it with acetone to swell it, and then this molecule benzophenone gets physically trapped in the silicone polymer, gets physically trapped. And so then when we come in and we put some sort of other polymer on top of the silicone and polymerize it under UV, the UV reactive benzophenone and whatever polymer is sitting there on the surface get entangled and make an interpenetrating network that cements everything together at the surface. So now we have a polyacrylamide gel, in this case polyacrylamide gel, very tightly bound to the surface of the silicone that we can manipulate and stretch however we want. Yeah?
So what is the difference between silicone and silicone wafer?
Oh, big difference. So a silicon wafer especially, is single crystal. So that is as beautiful and perfect and rigid as we could ask for. It's right next to diamond. But silicone, on the other hand, is a polymer that has silicon in it, but really whatever else goes into that polymer is fair game. So they tend to be very stretchy, very flexible. You can make them with very long polymer chains, or you can make them with some of the new optically clear silicones have shorter, more rigid chains, but they're very different materials. Other questions? Okay. And so this is what they look like when they're relaxed at null and strained 50%. All right, so they're strained a lot, which if you think about things in the body, nothing should be stretching 50% in the body. You're in really bad shape if things are stretching 50%. 10 is a good rule of thumb for physiological stretch. So given that, but when I started this project as a grad student and was like, I got it to work, the gel sticks to the silicone really robustly, it's really hard to pull off. The response unilaterally was, oh, but the strain doesn't transfer to the top of the gel. The gel is so soft that when you stretch the silicone, the gel is just gonna kind of sit there even if it's attached. So my next project became proving that the gel does in fact stretch. So in these last couple minutes, before we go to acknowledgements, why don't you brainstorm with your partner real quickly at least 2 ways to characterize the stretch of the gel. How much the gel is stretching when I stretch the silicone below it. And coming back to this error spread in the data question, what do you think are the sources of uncertainty in that type of calibration? Go. It's a good answer.
So you have one thing that's straight.
Oh, you can use
Like this, right? So
Sorry? Yeah, this was acrylamide in these studies.
When you use the UV, it will like polymerize it more?
It will, polymerize it? Yeah, it helps. It helps. So some people have specific UV initiators for acrylamide. I used the traditional chemical initiators, the APS and TEMID, the ammonium persulfate and TEMID, and the UV basically just helps create free radicals and speed up the polymerization.
First length
with the benzophenone, yeah, and so the UV also activates the benzophenone. There's a double bond to oxygen there, so when that gets hit with the UV, it sticks to whatever is nearby. All right, if you haven't talked about error in the calibration yet, please do that really quickly.
With your
No, that's definitely something that you want to keep in mind. A lot of these gels take a lot of plastic.
I mean, at 10% strain,
All right, I hate to do this because I can hear how awesome your discussions are, but I don't want to keep you past your food break. The way that we did it, did anyone come up with just measuring the gel itself? Yeah, we had, okay. Errors, what errors do we think? Depends on how good it depends on how good your tracing is. It depends on how well you can define the edge of it, and it also depends on how convinced you are that your sort of microns in real life to pixels on the picture, how good that conversion is. Okay, so there could be error in that conversion alone, and certainly in drawing circles around it is error prone. Did anyone come up with making marks on both surfaces and comparing them? Yeah, a couple head nods. What are some of the errors there? What problems do we run into?
Free body movements rather than straight.
Correct, right. So we could be detecting translation. Continuum mechanics is helpful there. We can mathematically cancel that out. But even in doing the math, you know, it's an analytical model, so it might not be matching real life perfectly. Other problems?
When you're tracking these markers as they come in and out of frame, you have trouble with recognizing which ones were from your previous frame and which ones were new.
Yeah, so you can see on the edges, right, as things shifted out of view entirely, we get very erroneous values. What I also thought Nick was saying is we just talked about how when you stretch stuff, it moves out of view. So what we could also be getting is particles popping in and out of focus that are throwing off our calculations. And then again, the same sort of image processing. Is our pixel to micron conversion actually good? How accurate are our analytical models? How much do we trust our particle tracking algorithms? These are all really important things to understand about the systems that we're building really across the board, whether it's a stretching system or whether it's some other kind of crazy system. You want to think about the errors you're introducing into your system just by looking at it. All right, and so then I did these calibrations and found that by and large, given a little bit of slop in the measurements, when you stretch the silicone, the gel's stretched and everything lined up really nicely. And then we also plated cells and then tracked how the cells were deforming. And so the cells, there was a lot of spread in the data, but by and large, they were not stretching identically to how the surface of the gel was stretching. So there was some loss somewhere in the cells that hopefully someone else will track down in the future for me. All right, so with that, when we're looking at stretchable cell culture systems, if you guys go to design them or if you're evaluating someone else's, we want to think about the stretching modality itself. So are they stretching it anisotropically in one direction? Are we trying to stretch it in plane so we can still look at it? Or are we happy to just puff it or push it out of plane and look at it later down the road? Is it mechanically driven by motors? Is it pneumatically driven by air or vacuum? How is this overall system operating? The two different systems that I described to you were a higher throughput one that was designed specifically with the idea of moving towards high throughput screening and looking at integrating with high throughput equipment, not necessarily compatible with a microscope. But on the flip side, I showed you that very pretty blue device, right, that was designed for the opposite. It was low throughput. You're only stretching one sample at a time, but you're directly over the microscope objective to be watching it real time. So design, by definition, depends on what you're designing it for. Right, so you get to make choices, which I think is one of the fun parts about engineering, is you're not stuck with observing what's happening, you're actually interfering and trying to control the systems. And then last, I also talked a little bit about designing a tunable layer on top of the silicone. And so with that, just a few design principles to take away. Again, if you do want to design these systems yourselves, you have to make good choices. So you can't have it all. It's impossible to optimize everything. So decide important parameters and then try to design the system around the things that are most important to you. As a side note, one of those things that tends to be important is how well does it integrate with existing lab equipment, especially if you're trying to propose something new to your PI. They're going to want to buy as little new stuff as possible. So think about what you already have around to build with and to integrate with. Keeping your cells happy is another important part of this. How's the environmental control going to work? How are you going to keep the cells bathed in media, or are you? If they're, how long is the system going to last? Do you need to have some sort of vascularization to clean out the cells and refeed them? Or is it going to be a quick study where you can just leave them and cross your fingers? And then last but not least, and we talked about this a fair amount with the gels, how are you going to characterize and calibrate the system? And what sort of errors are you introducing into the system? And can you design it in a way to minimize all of these things? All right, and with that, I want to thank everyone from my new lab at Florida, the GatorBAIT lab, that has been helping to build these new systems. Abby, my lab tech, has been with me from the start, and she's been great. And then Nick and Santi are here. I think most of you have met them by now. And of course, Beth, my former advisor, and Sean Palachek at Wisconsin and Viola Vogel at Zurich have been really helpful with my work as well. So with that, if there are any leftover questions, happy to take them now.
Thank you.
Yeah.
Time for questions? So I just want to make sure I understand it. So you take this whole setup and it's like in media, or how are the cells fed and stuff?
Yeah, so in the high throughput system, it's a tissue culture plate. So that's easy. They're wells, you dump them in media. You're correct to note that a flat silicone sheet, how do you keep those cells happy? You have at least two options, probably more, but the two that I've worked with the most is either permanently bonding some sort of retaining structure to the membrane, but now you're introducing new air into your strain field because you have some region that's reinforced, and you have to make sure that the well itself is stretching correctly. Or if you use mineral oil or Vaseline or something slimy, then you can put a retaining ring on top of the membrane that actually slides and will hold media on the cell quite well. And so those are the two ways that I've kept the cells happy in the stretching system so far, but I'm sure there are more.
So the cells are on the silicone, and the whole gel thing was just to demonstrate the change in stiffness. Is that correct? And you're not going to use the hydrogel for 3D culturing or something?
Yeah, no, we do. We do use the hydrogels for culturing. So if you have the silicone, and oh look, I even grabbed orange-ish and blue. All right. So if we have silicone and gel and cells, then you can put, if we have grease, then you can put a silicone ring around it and fill it with media. And then when you stretch down here, everything else here stretches, except this stays put because it just slides on the grease. Does that make sense?
Encapsulate the cells in the
I have slides if you want to learn about that. Yeah, so we have been using a very similar protocol to do stretchable 3D fibrin and collagen culture. cultures as well. But they're still in progress, and I didn't have enough time. Yeah. Maybe, yeah?
Given the figure that you have, are you, if I just extrapolate, are you thinking about introducing microfluidics to control the shear stress or electrical signals with electrodes and stuff like that?
Yeah, so, again, eventually we want to do it all without using a whole human. But one of the interesting design problems with shear and stretching is that if it's cyclic stretch, you're disrupting your flow. So trying to think about how to control fluid flow while you have essentially a vibrating membrane, that's a really complicated problem. It's a really complicated problem. And so actually we have a very new collaboration starting with Cecile Perrault in the UK at Sheffield, who is a microfluidics expert. And so we're working to start addressing some of those questions with her. And then there was maybe the last one, so we have time for drinks and snacks, but yeah.
I don't know if you mentioned the thickness of the gel and if there are viscous effects accounted for in your analytical models.
The thickness of the gel is about 100 microns. The speeds at which we've been operating to this point, we've not been worried about viscous effects, the strains, and the speeds, but certainly at a certain range of speeds and also for a certain strain. So strain magnitude and strain rate, you could certainly run into either nonlinear and or viscous effects. So nonlinear of stretching out the polymer chain too far, or viscous from the fluid that's trapped in the hydrogel. But we're making some small range approximations here and leaving off those terms. All right, thank you so much.